Humanity's ancient dream of defying biological limits is now reality. A tool exists that can rewrite our very genetic blueprint, offering cures but also forcing us to ask, what does it mean to engineer perfect versions of ourselves? Understanding CRISPR, or clustered regularly interspaced, short palindromic repeats, reveals a truly revolutionary genetic editing tool. This ingenious system originated as a natural defense mechanism, allowing bacteria and archaea to identify and neutralize viral threats by precisely cutting their invading DNA. Scientists ingeniously adapted this biological marvel, transforming it into the incredibly precise gene editing technology we know today. Its mechanism is surprisingly straightforward. A guide RNA molecule meticulously seeks out and binds to a specific DNA sequence, directing the powerful Cas9 enzyme, or similar, to that exact location. The Cas9 then acts as molecular scissors, creating a precise, targeted cut in the DNA. This innovation is truly revolutionary, boasting unparalleled precision, remarkable ease of use, rapid application, and significant cost-effectiveness compared to earlier methods. These advantages collectively make sophisticated genetic manipulation more accessible than ever, opening doors to previously unimaginable scientific possibilities. CRISPR is rapidly transforming diverse fields, moving beyond the lab into practical applications. In medicine, its therapeutic potential is truly exciting. Clinical trials are currently exploring its use to treat debilitating genetic disorders like sickle cell disease, beta thalassemia, and cystic fibrosis. By precisely correcting specific genetic mutations, CRISPR offers the promise of a one-time cure, fundamentally altering disease trajectories. It's also being developed to fight certain cancers by enhancing immune cells. Beyond direct patient treatment, CRISPR is an indispensable tool in fundamental biological research. Scientists utilize it to unravel complex gene functions, create accurate disease models for drug discovery, and develop rapid, sensitive diagnostic tools for pathogens, exemplified by its use in detecting viruses like COVID-19. Its impact extends to agriculture and biotechnology, where it's employed to engineer disease-resistant, more nutritious crops and improve livestock traits, paving the way for sustainable food production and a healthier planet. This diverse application spectrum underscores CRISPR's profound and expanding influence. The power of CRISPR extends beyond mere healing, leading us to distinguish between gene therapy and gene enhancement. Therapy corrects genetic defects to treat or prevent disease, addressing conditions like cystic fibrosis. Enhancement, conversely, aims to improve traits beyond typical human function, such as increasing intelligence, physical prowess, or altering aesthetic features. This pursuit of perfection immediately raises questions, as its definition is profoundly subjective and culturally influenced, making it a potentially dangerous and ever-shifting target. Moreover, complex human traits are rarely governed by a single gene. They're often polygenic and significantly shaped by environmental factors, making straightforward genetic fixes for desired enhancements scientifically unrealistic and overly simplistic. This crucial difference guides the ethical debate surrounding CRISPR's application. Navigating CRISPR's ethical landscape is like opening Pandora's box, demanding extreme caution. We must differentiate between somatic cell editing, where changes affect only the individual and are not passed on, and germline editing, whose modifications are heritable, impacting all future generations. This latter prospect presents truly profound ethical dilemmas. It raises the alarming specter of designer babies, fueling concerns about a new form of eugenics and the potential to exacerbate social inequalities if this powerful technology becomes a luxury exclusively for the wealthy. Moreover, we grapple with the unknown, the risk of unforeseen biological consequences from altering the human genome. There's also the persistent slippery slope argument that permitting therapeutic germline editing for disease could inevitably lead down a path toward non-medical enhancements, fundamentally redefining what it means to be human. These are critical waters to navigate. Shaping CRISPR's future demands responsible innovation. This begins with establishing global governance and robust regulations. International consensus and ethical guidelines are crucial to prevent misuse and ensure equitable access to its benefits. Alongside this, public engagement and education are vital, fostering informed societal discourse. This ensures decisions about CRISPR reflect broad societal values, not just scientific capability, building trust. Finally, we must critically define boundaries, asking who decides acceptable alterations? What are the long-term impacts on human diversity and identity? Do we possess the wisdom to wield this unprecedented power responsibly? CRISPR offers revolutionary cures but sparks profound ethical dilemmas on human enhancement. In your opinion, how should society define boundaries for editing our fundamental blueprint? Share your thoughts in the comments.